Yeah. So we're going to talk about extraction, amplification, and detection again. So step one, obviously you have to do the extraction. Um, so we have to get those nucleic acids out, and pretty much any specimen that can contain genetic material can technically be used. Some specimens are harder to extract genetic material than others. We use blood and buffy coat. Buffy coats, when you spin down blood, you can get the white layer of white cells. You get red cells at the bottom. You get the buffy coat, and then you get the plasma at the top. You can do whole blood. You can just do the buffy coat. Um, it just depends on the amount that you need and how much volume you have. So we work at a children's hospital, so we tend to extract from whole blood because their buffy coat tends to be very, very small because we get very, very small volumes of blood. So we can use solid tissues, and you would mince the tissue up into small pieces and then digest it. We could do viruses, bacteria, fungi, anything from like a plate potentially. Um, dried blood and plasma. So dried blood would be on those little spots, like on the. You guys ever like see a newborn screen when they drop the little blood into like the cards and the blood absorbs into the cards and the circles and everything? Did you ever see that or no?
Yeah, we get those sometimes at CCL.
Yeah, so you can extract DNA from dried blood. Plasma, bones and teeth, hair follicles and hair shafts, formalin fixed tissue. We actually use formalin fixed tissue for Bartonella Hensleyi detection in our lab. You could get it from soil, um, lavage fluids, so like BALs. Um, you can get it from saliva, urine, amniotic fluid, CSF, and stool. So like when you extract from formalin fixed tissue, there are some extra steps. Also with stool, there's some extra pre-treatment steps. Again, you got a lot of extra stuff that you need to get rid of and break down. So there's extra pre-processing steps um, in the extraction process. So the general goal of nucleic acid extraction is no contamination of the specimen. You want to ensure that whatever your specimen is, it actually is. You don't want to cross-contaminate with your cells or with another person's sample. Um, the control, you don't want to contaminate at all. So you want to ensure that the DNA you are getting is the DNA of interest from your specimen and nothing else because it's very sensitive. You get one extra copy, two extra copies in there that shouldn't be in there, and you're not necessarily detecting the DNA from your specimen. So we need to lyse the membrane. Um, but we also want to, through that whole lysis process, when we're breaking down the cell membrane, we don't want to damage the nucleic acid. So the specific chemicals that are in the extraction process are also there to, even though break parts of the cell down, keep other parts intact. And then we want to purify the target material from all that other junk that got broken down. A variety of techniques can be used, and largely depending on specimen type. You can centrifuge peripheral blood either to get plasma or to get that buffy coat. A puffy coat, yes. Thinking winter, I almost said puffy coat. You can crush or mince fresh tissue. And then a variety of chemicals and or enzymes are employed to lyse cells, often available as commercial PCR reagents. So everything that we do in the lab currently, all of our extraction methods are automated and we buy kits to do it. We used to do manual extractions. Cytogenetics took over our manual extractions once COVID hit. There's still kits that they purchase to do it, but they have to do lots of manual processes in order to, so they're adding each reagent, they're spinning stuff down, then they're adding another reagent and they're spinning stuff down to purify it, opposed to using these automated extractors that use magnetic glass particles and stuff. So organics are such as phenol and chloroform can separate different layers. Inorganic chemicals such as detergents, EDTA, acetic acid, and salts. We kind of talked about EDTA and how that helps preserve the nucleic acids yesterday. And then solid phase, so DNA is immobilized in a solid support, so that could be on a solid column or magnetic column or glass beads. So there's lots of different methods. So specimen collection, you can collect bone marrow or peripheral blood. We use and collect in our lab, we collect EDTA lavender pink caps. We don't use acid citrate dextrose, which is the yellow cap. I added to the slide for tissues. We can either use fresh tissue collected and saline processed immediately. You want to process fresh tissue immediately because it can start breaking down, and all of those enzymes and stuff as it breaks down are going to degrade your nucleic acid in there. So you want to, if it's fresh, you want to process it as soon as possible. You could do fixed or frozen. So if it's frozen at -70, that's fine. You're stopping those enzymes from breaking your cells down because, you know, -70 is really cold. And we know that, you know, enzymes act at a certain temperature, and -70, they're very, very inactive. You can fix with other things, um, like formalin, but we also use FFPE, which is formalin fixed and paraffin embedded. So not only is the tissue formalin fixed, but it's also paraffin embedded. So it's those little like wax blocks, essentially, and that the tissue is embedded in so that you can get like small slices of tissue. So you want to embed it and fixate it to keep it rigid when you slice through it. So you can get really, really thin slices. We use like 5 micron slices, and 5 microns is very, 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 very thin. So a solid phase extraction using automation. So you put your cells in suspension, you lyse it, so then you get the DNA in the aqueous solution using like acidification. So you have acids and stuff, you spin it down, like you get the cell debris down there. You have a low pH, so you absorb the DNA, you wash it, and then you elute it. So your magnetic glass particles are stuck in here, and you elute it, and you're left with just your DNA. Sometimes you do get magnetic glass particles that come out into your elution. You want to spin your final elution tube down, so because if you put magnetic glass particles into your PCR amplification, it can inhibit PCR. So you want to ensure that when you're finally getting that DNA, you spin this tube down to get any extra debris down at the bottom. If we're extracting RNA, because RNases, things that break down RNA, are ubiquitous or everywhere, you really have to be careful to minimize contamination and exposure to your sample to RNases. So there's this spray called RNase Away, which essentially breaks down RNases so that you can process RNA without it degrading. So your bench and equipment must be designated as RNase-free areas. You need to have gloves and wiped with RNase cleaning agents or RNase Away. Disposable equipment must be certified RNase-free and rinsed with 0.1% diethyl pyrocarbonate or DEPC. So because RNases are ubiquitous, you need to ensure that all of your supplies are DNase RNase free. So we order just DNase RNase free supplies for everything that we do. Even though we don't particularly extract RNA except for COVID's, everything else is DNA, we still have everything being an RNase free environment. So reagents must be certified also to contain a very, very, very they must be certified to be RNase free, and then they have to contain a very, very small amount of DPC. You have to test the reagents with RNase detection agents, and then you use organic and solid phase methods. Types of RNA, there's lots of different RNA types. So depending on what you are trying to study, you could be isolating different kinds of RNA. So you have messenger, ribosomal, transfer, heteronuclear, small nuclear. You can have double-stranded RNA and microRNA. So heteronuclear RNA is within the nucleus and it's pre-mRNA. And other RNA transcripts, they don't move to the cytoplasm. After they're processed and converted into mRNA, then they can move to the cytoplasm. Small nuclear RNA is small RNA molecules that are found within the splicing speckles and caudal bodies of the cell nucleus in eukaryotic cells, but most of the time what most people are looking at is mRNA. They want to know what is actually getting coded to leave the nucleus and to get finally transcribed and translated into a functional protein or whatever. So most people, when they're studying RNA, they're extracting mRNA, and that's why there's an asterisk there. So a way that we can isolate mRNA is you have this column. It can either be a magnetic bead or a column, and they have these poly T tails bound to them because mRNA has a poly A tail. So all your mRNA will bind to this bead or column with this poly T tail that is on it. So that's how you can target just your mRNA that is coming out or that is in your extraction. Does that make sense? And that's just something that is all mRNA has. All mRNA has a poly A tail just based on its processing. So step 2 is amplification. There's some YouTube videos here that show PCR. I'm not going to show these. We can maybe come back if we have extra time, but yesterday I went really over, so I don't want to do that tonight. So we'll come back if we have time. You can watch them on your own as well.
We're fine.
What?
It was fine.
I know, but I still feel bad. I want to, if they can watch a video on their own, I want to make sure I am respectful of everybody's time. So PCR primers, we talked about primers a little bit yesterday, and I did want to add a picture here. So I'm gonna add a picture because I'm a visual learner. But primers are single-stranded and they're 18 to about 30 bases in length. You don't want them too short because they should be specific to the gene that you want to target. And the shorter the sequence, the more likely it is to be found in another organism or another region of the genome that you're trying to target. So it might bind somewhere that you don't necessarily want it to bind. So, but it shouldn't be too long because then it's going to have trouble finding where it needs to bind, and the longer it is, if there's slight mutations or something's not highly conserved in a specific region of the DNA, you could have variations and get no binding when you want binding. So you really try to optimize your assay, and it should be complementary to the sequences flanking the region to be amplified. So you want to develop a forward primer and a reverse primer. So you want it to flank both ends of your gene because you only want to amplify that gene region of interest. You don't want it to keep going and going and going and keep amplifying and extending the entire genome after that initial probe and primer bind. So you want short, concise sequences. That way you can cycle through very, very, very fast. That's why you can get, you know, you only have extension steps that are 30 seconds or 45 seconds long, because it can actually go very quickly through that process. And then the distance, again, primers determine the specificity of the PCR reaction, and the distance between primer binding sites will determine the size of the PCR product. So if your primers are very, very close, you're going to have a very, very short PCR product. So your extension time is going to be much less, and your annealing time and everything else like that is going to be much less. But if you want a very large PCR product, obviously your primers will be farther apart from each other. And then you'll need longer extension times. So yeah, does that make sense so far? Are we good? Questions? Okay. So the PCR recipe. So typical reactions have specific compounds. So you can have obviously your primers, and this one says, okay, 0.25 millimolars of each primer. Again, that can be changed, and you optimize your ratio of primers and your concentration of primers based on a lot of different things. Your limit of detection, the amount of DNA that initially goes into your sample. So there's a ton of different reasons to optimize that concentration. It directs DNA synthesis to the desired region. You have obviously your DNTPs, which are DATP, CTP, GTP, and TTP. Again, the longer the sequence that you're using to target, obviously each cycle will then use more nucleotides. And the shorter the sequence you're targeting, obviously the less nucleotides you would use. So again, this is optimized based on the size of the sequence that you're targeting. You have 50 millimolars of potassium chloride, and you want those in there for optimal hybridization of primers to the template. The template is the genome of interest that you're targeting. We call it the template because that's what we're copying. You have 10 millimolars of tris at a pH of 8.4. That's alkaline pH. We talked about that yesterday. And that is optimal for some of these enzymes to do their reaction, like the Taq polymerase and stuff. You need 1.5 millimolars of magnesium chloride. And again, this is a divalent cation, and it's required by the enzymes in there. Again, the more enzymes you have, obviously, the more magnesium chloride you'll need. But also, we looked at how the concentration of magnesium chloride affects melting temperature. So if you're doing a melt curve analysis, that can also interfere. So again, all of these things can be changed, but they're very, 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 like you just have to set up your assay so that they're the same every time. And then 2.5 units of polymerase. And again, you're optimizing that. You want 10 to the second to 10 to the fifth copies of template. If it's something that you can control. So we nano drop a lot of our genetic studies, and we dilute. After we extract, we determine the concentration of DNA, right? So then we dilute and we control how much DNA goes into those assays based on our testing needs. But for infectious stuff, you don't know how much is there. So when we extract DNA, we don't nano drop it. We don't detect the concentration because we want to put, you know, what we can and what we've optimized, the amount of volume that we've optimized for our assay to put in, and hopefully something's there that we can detect. Does that also make sense?
Yes.
Assuming silence. Thank you, Grace. I appreciate that. I don't know
No
if
problem.
everybody's sleepy. Do I need to be more energetic and keep you guys awake? Because I'm sleepy. So let's go. Like, I'm gonna make you participate. So PCR, remember 3 steps of PCR are denaturing, annealing, and extension. That you should memorize. Denature, anneal, extend. Denature, anneal, extend. You have to, those are the 3 processes. You just have to memorize it. So if you get anything out of this, know that. Okay. The PCR product is called an amplicon because that's what you're amplifying, right? That's your amplicon. And the number of amplicons is 2 raised to the nth power, and the n is the number of cycles. So if you go through 40 cycles, right, because each cycle should give you for 2 copies of your original. And if you go through 40 cycles, you have 2 raised to the 40th number of amplicons. Again, know that the temperature of denaturation is the highest temperature, right? You want to separate those strands. So you have a temperature of 90 to 96 degrees Celsius, and that's anywhere from 20 to 60 seconds. The longer your strand, the longer this denaturation time because you have more hydrogen bonds to break. Also, the higher the C to G ratio, the longer this time is, the higher this temperature is going to be, right? Because there's more bonds to break. Annealing, you cool it down, right? You're bringing it down, things can settle. They're not vibrating, going crazy, right? Think about the kinetic theory. The hotter you are, the faster your particles are moving, so your DNA can break, and then you slow down, your particles are slowing down in there. You cool down, you can meet up again, chill, hang out. And then we have extension, and then that is from 68 to 75. And that's in the middle, right? So you can finally, like, your enzyme is warm enough to do its work. That Taq polymerase is like, yes, this is my happy temperature and I'm good to go. And again, depending on how long that sequence is, depends on that time that you're going to set this step for. The shorter the sequence, the shorter the time. So yeah. Again, denaturation, it's separating those 2 strands. Here's all your primers. This dark purple region is that region to be amplified. So you have one forward primer, you have a reverse primer. So they're going to bind on opposite ends of this gene. This first cycle is going to extend beyond, right? You get this extension beyond your region of interest in both directions. But then when you go on to the second cycle, I'm going to include another picture because I want to show you what the next cycle looks like. So when these 2 strands separate, right, you are going to have a primer bind right here to this spot. Oh, and right here to this spot. So then when it copies, you will only get short little segments, right? So you will have the majority of short little segments, but after you get like your 40 cycles. But each time you have a denaturation, you get this strand exposed again. So you end up having like quite a few like you have your original strands and then you have some mediocre length strands, but then most of your amplicons will end up being very, very, very short. So yeah. So I will add another picture to show you what that looks like. PCR automation. So PCR requires repeated temperature changes. So thank God they've come up with things called thermocyclers because it is very, very, very hard to manually go, okay, 10 seconds in this ice bath and 40 seconds in this heat block and, and like manually change things. That's crazy. So an instrument does it for you. Thank the Lord. So the thermocycler changes the temperature in a block chamber and it holds the sample steady and it just does its thing and you just walk away and it's beauty when it works. Thermostable polymerases, which we call Taq polymerases, are used to withstand repeated high denaturation temperatures. So does anybody know where we got Taq polymerase from? Where do we find an enzyme that can withstand the high temperatures of the denaturation step.
Was it from like a bacteria in a geothermal place?
It is. It was from bacteria in a hot spring. So it is optimized for, it's like, yes, I live in a hot spring. I am Gucci. I know how to withstand all these high temperatures and I can do my job still. That's why we can't use our own, because our own enzymes would denature at that temperature. So again, you gotta love science. Beautiful. Okay. It's a great thing. Great thing. Anyway, so PCR results. The PCR product should be of expected size. We don't run gels anymore, but I guarantee you need to know how to read a gel. On my exam through ASCP, I had gels that I had to read on my test. So you should know how to read a gel. So we always have molecular weight markers. Some labs put them on both ends of the gel just in case for some reason the gel, like, you know, is crooked and runs faster on one side than the other. Usually if that happens, you need to repeat it. So it loads very, very, very evenly, but just in case they do, maybe your reagents, your agarose, when you were dissolving it and you poured your gel, you didn't dissolve it all the way. So putting molecular weight markers on both sides of the gel, make sure that your agarose is of equal concentration throughout the whole gel, which is also very important. So in this picture, though, it only shows molecular weight markers on the one side, and that's fine. So these you purchase from a company, it gives you known molecular weights that are in the gel. So maybe this is 1,000 base pairs, and down here it's 50 base pairs, right? And they sell different molecular weight ladders based on the size of the genome or the target amplicon that you're trying to monitor or detect, right? So you know that yours should fall somewhere in this specific weight ladder that you pick out. So the PCR product should be of expected size, and you determine that based on your weight marker, right? No product should be present in the reagent blank. This is your reagent blank lane, right? Your primer dimers, so when your primers bond to themselves, and you get these things called primer dimers. So those will be really small though, and those will run really fast, and those will be at the end of your gel. So that's okay. But no product besides that should be in your reagent blank because nothing should be amplified. But your primers are still going to be in there, but they won't bond to anything to amplify. So you'll still see them though. Misprimes may occur due to nonspecific hybridization. So if you see something, right, this is a misprime. You have nonspecific hybridization in here. So it's not of your desired length, but obviously you've seen something. So there's something else in there. Maybe you need to redesign your primers if you're seeing misprimes a lot. Who knows? Maybe there was contamination in this one of some other type of DNA that also bound to it, but you never know. And primer dimers may occur due to hybridization of primers to each other, which I already said. I got ahead of myself. Sorry. So PCR contamination. Any molecule of DNA containing the intended target sequence is a potential source of contamination, including your DNA. So human DNA was our source of contamination when we had our issue just a few weeks ago in the lab. We had to bleach everything down to break down any extraneous sources of DNA that might have been on the equipment handle, in the fridge, like because people were touching things without gloves. So we had to get rid and bleach everything down. And the most common contaminant is residual products from a previous PCR reaction. So after every time we set up a reaction, we clean everything. You open a tube and you could potentially have aerosolized DNA spewing out into your little hood box or into your instrument. We don't open any DNA amplification tubes in our detection room. so that we don't have residual products from a previous PCR reaction spewing out everywhere. We do have a light cycler that uses glass capillaries, and occasionally the glass capillaries break. In the next 2 years, though, it will be sunsetted, and we will hopefully get a whole new instrument that doesn't use glass capillaries anymore, and we will never have this problem, and it will be amazing. I cannot wait. But yeah, so you want to minimize the risk of especially breaking one of those tubes, because that's where the PCR reaction happened, and you have millions to billions of copies of that gene of interest that are just spewing out in your instrument, and you're like, oh my god, I need to clean this up. Headache. So yeah. Contamination controls, we have airlocks and positive airflow. We have PCR hoods with UV lights, and we use a lot of bleach. We make it daily. So we do have airlocks. We have separate rooms for each setup piece state. We have separate rooms for each setup technique, and when you rotate through molecular, they'll talk about all that. Some of you already did your rotation. So you saw PCR 1, you saw PCR 2, and you saw the detection room. So automated PCR is PCR with colorimetric detection products. Samples are amplified and products detected automatically after a PCR reaction. And we use this for infectious disease applications. We don't do Neisseria or tuberculosis, but the last person who did this PowerPoint was from Summa. So do they do that at Summa? Oh, Jen, you're from Altman. Never mind. Where are you from, Jennifer?
Yeah, I'm Altman, but Mary's
Yeah.
here. She might know about Summa.
That's okay. Anyways, there's lots of other diseases that just not Akron Children's.
I wasn't paying attention. I just heard my name.
I was asking, the last person who did this PowerPoint was from Summa, and they have Neisseria, tuberculosis, and chlamydia. They do PCR for those. Is that what they do? We don't do that here, so I was just asking.
Do some of that, yeah.
Okay, yeah. So we haven't set up any of those assays for in our specific lab, but some of that is on the automated platform in virology, not in our lab though. So automated PCR, real time and quantitative. And then we also have reverse transcriptase. So you have to be careful on whether, because people use RT-PCR as real time or reverse transcriptase. So remember, reverse transcriptase PCR starts with RNA as your product. Real time PCR, it doesn't matter what it starts with. It's just saying that it can detect how much DNA or RNA product that you have in your tube live after each cycle. So standard PCR with an added probe or dye to generate a fluorescent signal from a product, and the detection of signal in real time allows quantification of starting material. So we do quantify CMV, EBV, and BK viral loads in some of our HEMOC kids. So we put a standard, we PCR a standard. So we have a high analytical measuring range sample, a low analytical measuring range sample, and we have a standard. So we assign the standard the value of viral copies. Like let's say we put in 30,000 viral copies. Wherever that sample falls on that standard curve as it gets generated is 30,000. And it uses that standard to quantify viral loads in all of your other patients. So we don't do anything else besides CMV, EBV, BK at the moment, but we're bringing up adenovirus as well. So that's something that we're currently working on. Again, they're performed in specialized thermocyclers with fluorescent detection systems. There's another YouTube video. Again, we're going to skip that for now, but we're going to come back if we have time. So there's lots of different PCR detection methods. So branched DNA assay. So we have branched oligonucleotides. So you have signal amplification and molecular diagnostics. So you have a capture, you have a plate, you have a capture probe, and then you have your capture extender, and then that actually binds to your target DNA. And then you have a labeled probe and a labeled extender. So this is a preamplifier. So you don't, it's not actually, it's not necessarily amplifying quote unquote like your DNA because your DNA is captured to the surface. It's actually amplifying the amplifier. So your signal is then your signal is increased that way. So if you have, oh, let's say 50 copies of DNA bound to your plate, you have these labeled extenders that will bind to here, and then your amplifier signal, as it binds, it actually gives off a signal. It will not give off a signal if it doesn't bind. So the more DNA you have bound to the plate, the more signal will be given off as this amplifier binds to it. So we can detect in a number of different ways. We can do electrophoresis. Again, it's kind of outdated at this point, but still people do it. And it's the movement of molecules by an electric current. And nucleic acid moves from negative, the cathode, to anode, which is the positive pole, because DNA is negatively charged. So when DNA is applied to an agarose or polyacrylamide gel, its migration under the pull of the current is impeded. So remember, it slows down through the gel, and then the different concentrations of gel you have can make it faster or slower through its migration process. The movement of molecules is impeded in the gel such that molecules will collect or form a band according to their speed of migration. And the concentration of the gel buffer will affect the resolution, so the separation by size. So resolution means the separation by size. So you can see that this is a 2% gel, a 4% gel, and that is a very, very blurry, I don't know, 8% maybe. So you can see that these bands move much further, right, and much quicker in a 2% gel. But in the 4% gel, it depends on what band you're targeting. So if you don't care about these small bands and they're not of interest, right, you can get a higher definition, right, if you concentrate the gel a little bit, as long as you don't also care about these bands up here, because obviously you can't distinguish these bands. They're moving too slow because they're too big. So you choose your concentration of gel based on what bands you're trying to target. Again, we have different buffers. And they carry current and protect samples during electrophoresis. And we talked about TBE versus TAE buffer the other day. And then RNA, we use sodium phosphate buffer or MOPS. We don't do RNA and we don't do gels anymore. So gel electrophoresis can have either a horizontal or vertical format. Again, you load your samples into these sample wells with the dye. I've run both, but horizontal is typically agarose, and that's mainly what I've done. But vertical is polyacrylamide, and I've done this, but not very often. And then we use combs to make the wells so that we're putting our sample into, and there's different size combs to have different amounts of samples that and depending on like the size of your gel, like there are gel boxes that are super wide, there are some that are really skinny. It depends. Like they make different gel boxes to suit different needs. And then you use the proper gel concentration for sample size range, use the proper coma and gel size to accommodate sample number. You run your molecular weight marker for known fragment sizes. You mix the sample with a tracking dye. We tend to use bromophenol blue. And that way you can actually track to see where your sample is in the gel. If your dye runs off the gel, chances are you lost your sample as well. So if you run it too long, it will literally just run off the gel and it'll be in your buffer and you have to start over because it's gone. You can't analyze it at that point. It's always better to run it a little shorter and realize, oh, it doesn't look like. I would check on it all the time. You never know. You don't want to lose it. I've had that happen before too. So you have to pipette the samples into the wells that are made and connect it to a power supply. You confirm that the current is passing through because there's little bubbles that form on the anode and cathode because it's splitting the water into oxygen gas and hydrogen gas. So you'll actually see bubbles. That's how you know that there is a current. Again, we're going to skip the video, but yeah, we might have time. Detection, assessing yield of extraction. So ethidium bromide and cyber green are for DNA. Ethidium bromide, people try not to use anymore because it's a carcinogen. So cyber green is a little safer. And then we can use it for RNA as well, but cyber green 2 is for RNA. There's silver stain. There's band densiometry for quantification, so you can actually determine the density of these bands. So obviously the more dense a band is, the higher the concentration of DNA or RNA that is in that band. The lighter the band, right? This is a very, very light band right here. Obviously it has less DNA in that band. So you can actually determine and quantify how much DNA you have in there based on the density. And then detection, assessing yield of extraction. You can use fluorometry, so it accurately measures intact DNA. We use Hosch dye, and then it can measure RNA with cyber green 2, but contaminating DNA won't interfere. So you can see that it will bind to your intact DNA and say, okay, I have 1, 2, 3, 4, how many cells or whatever that you are, or probes or primers or and then you measure the fluorescence intensity of it. You can use spectrophotometry. So this we use as a pre-analytical step before we do our PCR for our genetic assays, just to quantify what we're putting in there. But you can also do this post-amplification as well. If you don't have like fluorescence monitoring, so you can measure your 260 and your 260-280 ratio to determine the purity of it as well. So again, we look for, it says 1.6 to 2 for DNA. We look for 1.8 to 2, and then we want above 2 to 2.3 for RNA. So we want 1.6 to 2 for DNA. Remember, if it's lower, if our 260 to 280 ratio is lower, we have protein contamination, and if it's higher, we have RNA contamination. Oh, yeah, that's it. Was this